Good evening to all of you and good morning to all of you. It's great that we are having the Meolive sessions. It's great that Giovanni and Jeroen has initiated this wonderful platform to discuss and collaborate with several meiobenthologists globally. It's a great effort. I really have hats off and salute to these two wonderful guys and all my colleagues on the Meiolive program. Now, the presentation of the day, which I'm just doing today, is the diversity and community ecology of Meiofauna from the Arctic fjords. The background and significance of this presentation is most of this presentation is part of this study, is actually part of the Indian Arctic Research Program, which way back started in 2008 in the Indian research station in Himadri in the Svalbard, near to the shores of the Kongsfjord region. And the important part or the concept of this Indian Arctic Research Program was to study the impact of climate change, how climate change and the thinning of ice, the erosion in ice and other atmospheric and biological changes that's happening in the Arctic fjords. The Kongsfjord, the fjord ecology has been taken as a model for predicting the climate change, along with several other, more than 15 countries are part of the International Arctic Research Program, and India is one of the signatories, and Himadri is, you can see Himadri up there on the right side at the top, and India was part of this international program, and we came into this international program in 2008. And as part of this, my university, where I represent the Department of Marine Biology, Microbiology and Biochemistry, Cochin University of Science and Technology, has been selected for, we have been continuing with this program from 2011, 15, 16, 17, 18, and almost 19. But this year we are not in the process because of the COVID issues. Now, because there are many studies which have been done on the meiobenthos from the Arctic fjords, especially the Kongsfjord, the Crossfjord, and the Svalbard region. Now, the concept of this is why do we need to do these studies? Because the need of the biodiversity changes, which is definitely linked to the climate change, has been one of phenomenal in Arctic, which is identified as a hub of for the climatic change phenomenon. Now, what we looked into was the community pattern of meiobenthos, both in the inner and outer glacial fjords during this study. The study was mainly focused to test the hypothesis whether there is a significant difference between the meiofauna abundance distribution and the community ecology for major groups between the inner and outer fjords, which were basically influenced by definitely the glacial inputs, the oceanic inputs from the Atlantic Ocean, and also the riverine inputs from the nearby areas. So the objective of this presentation, see, this is a continuing study. I'm just putting you some of the thoughts and some of the findings which you have come at this is still at the preliminary stage. And a few publications have come out in these lines, but still more are in the process or on the anvil. Now, the objectives which I am going to is defined in this presentation is to analyze the composition, abundance, distribution of meiofauna and the major meiofaunal groups, along with the taxonomy, feeding guild analysis, and the carbon potential of anti-sequestration efficiency of some of the nematodes, forams, foraminiferans, and the taxonomy and the community carbon potential of foraminiferans. Then third objective is identify the key environmental drivers or variables which is controlling the meiofaunal distribution and diversity in the Kongsfjord. We basically did, our research continues in the Kongsfjord and Crossfjord, but what I'm going to present here today is the Kongsfjord. See, this is just an idea on the Kongsfjord and region. We have selected, you could see the Kongsfjord here, and you have selected 1 to 7 locations, which is marked at a depth of 76 meters, 80 meters, 40 meters. 1 to 3 is 76 to 40 meters. Now, 4 to 7 is 303, 300, 230, and 244 meters depth. And I have just shown you the latitude and longitude. All for information. I think some of our colleagues on this Meiolive program, they may have already been doing, they're still working on it. But the interesting thing is I don't find much of meiofauna studies beyond 2003, 2004, or 2005, which is still importantly published. So the latest study, what we have been doing is adding to the information and updating many of the literature and biodiversity status in relation to climate change issues in the Arctic Kongsfjord. Kong's Fjord is a border area between the Atlantic and the Arctic biogeographic zones, influenced by, as I said, the Arctic and the Atlantic water masses harboring a mixture of Arctic flora. As I said, we have collected samples from 7 locations at 7 different depths, divided into 2 subsets. The inner fjord, please understand, it is the stations 1 to 3, and outer fjord is the 4, 5, 6, and 7. The inner fjord was definitely influenced by the glaciers, and the outer fjord was mostly had the oceanic influence from the Atlantic Ocean. Of course, we all know that meiofauna is very, very important, and this meiofauna, it's very relevant in terms of its adaptability in food availability, microhabitat heterogeneity, and its contribution to bioturbation. And this is some of the methodology we are following. I've just given you for information. Now let me come to the meiofauna distribution during. So I'm just going to present you the meiofauna studies in Kong's Fjord during 2011 and 2015. So I'm just making a comparison. What are the changes? What's happening between the 2011 and 2015? Now, if you look at 2011, what we mainly see is meiofaunal abundance was high in the inner fjord, which is mainly contributed by 37% forams. You could see here 37%, 54% was mostly by nematodes. You could see high meiofaunal abundance was observed in station 2, possibly in the lower depth stations, followed by 1 and 3. That's the lower depth stations. Then nematodes were the dominant taxon in all these stations, representing about 54% of the total meiobenthos abundance. Also, the total biomass contributed by meiofauna during 2011 study was around 6445.63 microgram per 10 centimeters square. Biomass of meiofauna was abundant in station 2, that is the lower depth stations, and lowest also in the another station 4, which is also having 783.53 micrograms. You could see I've graphically given on the right side the presentations. Now, if you look at 11 also, the species richness, you could see that there was a species richness, the Shannon index of, and also the evenness and the species dominance was higher towards the inner fjord, whereas the outer fjord showed considerable differences with compared to the inner and outer fjord. The MDS plot also, MDS plots, if you looked at the MDS plot, you could see that here almost similar. You could see that the similarity in the abundance of meiofauna in the inner fjord was more pronounced at the lower depth stations 1 and 3, that is 93%, and around for 5 and 7 it is about 96% in the outer fjord. Overall similarity in the stations was almost about 80%. We had similarity here. This is depicted by the multidimensional scaling ordination, and also we could see the cluster dendrogram plots. If you look at 2015, you could see that this was represented mostly by the abundance was represented by nematodes, the nematoda, foraminifera, ostracoda, harpicticoid, copypoda, chinorhynchs. Then you had oligochaeta and Tintinida. The percentage abundance, if you look, you could see that the average numerical abundance in terms of percentage in the inner and outer fjords are almost similar. See the difference compared to the 2011. Higher meiofaunal abundance was observed in station 3, followed by station 1 and 5. Then, of course, nematodes, the forams, and the dominant taxon here. Total biomass contributed to the meiofauna during the 15 study was around 3150.336 microgram per centimeter square, 10 centimeters square. Biomass of meiofauna was abundant in station, the lower, that is the low depth station, station 4, and also the another lowest in the station 6, the high depth zone, that is around 328 microgram. If you look at 2015, the richness and the evenness and the Shannon index, you could see that the high species richness we could see during the inner parts of the fjord. Here also the similarity in abundance of the meiofauna was highest in station 1 and 5. And also if you look at the multidimensional scaling image and the results, we could see that here it is 60% similarity, where we had almost 80% similarity in 2011. So that difference you could see between the two. If you look at 11, the two dominant, we also looked into the two dominant groups or the taxa, which mainly represented by nematodes and the foraminiferans. We could see that we looked into the species characterization. I'm just giving you running through the species structure. Outer fjord had high numerical abundance and biomass of nematodes compared to the inner fjord, where you look for the nematodes. It's just the opposite of the total meiofauna. Higher abundance of percentage abundance was observed in station 3. the low depth zone, followed by stations by the locations 4 and 5. Nematode biomass was possibly very high in station 6, followed by 4 and station 3, the low depth zones. So you could see here at the graphically, you could see the biomass, the changes in the biomass station-wise or spatial variation in the biomass here. If you look at station 11, the nematodes of station, during 2011, we could see that the richness, the Shannon index was higher in the inner fjord, and the species dominance was also high in the outer fjord. The species richness did not show any major variation in the fjords. The MDS ordination plots also showed that the similarity in abundance of nematodes was most pronounced for stations 2 and 5. That's about 65%, which depicted here in this graphical plots. MDS also showed a 40% of all the similarity which had for nematodes in all the stations. Percentage abundance of nematodes, if you look for 2015, we could see that the outer fjord showed highest abundance of biomass of nematodes. And here you can see the spatial wise, that is the station wise 1 to 7. You could see 850, nearing 900, 850, 890. And highest abundance was observed in station 4, followed by 1 and 5. Nematode biomass was highest in station 4, followed by 7 and 6 in the Kongsfjord. Now, if you look at the diversity and similarity indices during 2015, we could see that the similarity, the species rich evenness was quite comparatively higher in the inner fjords as far as nematodes were concerned in 2015. Similarity in abundance of nematodes was maximum if you look at station 2 and 5, that's about 80%, and 40% similarity was observed in the nematode abundance between stations. The cluster, you could see the MDS ordination which is projected here, 2015, and this depicts the 80%. Now, if you look at the free-living nematodes, marine nematodes of the Kongsfjord region, we could see the highest percentage abundance in the first column, the low abundance species here, and the very low abundance on the right side, where you could see that the Sabbatiaria, which is a good colonizer, showed the dominance in the disturbed sediments, that's possibly in the inner fjord. Tertialinga communities were generally associated with the silty sediments in the outer fjord. All the species observed were truly marine in nature, and most of them were being euryhaline and few of them being polyhaline. You could also see the presence of Dorylomopsis species, Mesconium, then Sabilaria, Tertialinga, so many species. But very low abundant species I've given in the last column is very clear for you, all of you. If you look at the free-living nematodes for 2015, you could see that 32 genus of nematodes representing 15 families were taxonomically identified from this. Here we could see that the highest abundance we could see, that's the Dorylomopsis, Sabbatiaria. Then the low abundance you could see in the central column, and the very low abundance represented by the last column. Now, if you make a comparison between 11 and 15, what the nematodes suggest is that the faunal assemblage of the Kongsfjord was represented by 45 genus of species, and whereas in 15 it was represented by 32 genus. Dominating species is Dorylomopsis, Tertialinga, Longicordata, Sabbatiaria, then Mesconium. Then regarding the dominating in 2015, almost overlapping, almost Dorylomopsis, Sabbatiaria, Tertialinga, couple of species, which you see the percentage variability is also in Dorylomopsis was 64% in 2015, whether it is here, it's only 45%. So many of the less abundant nematodes were observed, like for example, Trobilla species, then Enoploid species, then you had Meton, Colamus species, and so many Neotobrilla species and Quasibrilla species, and all were less dominant, were represented during 11, whereas the less abundant nematodes were not observed during 2015. Then, of course, chromodorids and aerolomids were abundant in the inner fjord. Only chromodorids were abundant in the inner fjord here for 15. Then enoplids and monhysterids were abundant in the outer fjord. Enoplids were abundant in the outer fjord. Sabbatiaria, which is a good colonizer, showed its dominance in the disturbed sediments in the inner fjord. Whereas Sabbatiaria showed the dominance both in the inner and outer fjord during 15. Now, if you could, we could do a little bit of feeding guild analysis of the nematodes we collected in 2011. What is I just come to the outcome of what is the overall outcome. This is also a published work. we could see that we had also selective feeders, non-selective deposit feeders, epistratic feeders. Then, of course, predators of omnivores, epistratic feeders and deposit feeders dominated in the outer fjord. Predators, of course, were dominated in the inner fjord. So what we always concluded from that 2011 was that you could see that around an example of epistratic feeders like Sabiteria punctata, Sabiteria ornata. Deposit feeders include Tertilinga longicordata and Spherolemus gracilis. Predators like Thoracostoma species, Viscosia species, and nematodes. The tail plays an important role in locomotion. The cleavate and the long filiforms also represent the tail forms in the outer fjord, and the conical type in the inner fjord, regarding the long cleavate and tail shapes. Cleavate tails were dominated in the sandy regions of the inner fjord. Conical tails were prevalent in the sand and muddy sediment, and long filiform tails were dominated in the clay sediment in the outer fjord. So if you look at the feeding guild, you could say that the 20% we could see domination by selective, you could see by it is dominated by selective deposit feeders. That is 1A, or followed by, you could see 22% was non-selective deposit feeders, and the highest was, of course, the predators or omnivores. And now, if you look at the feeding yield for the 2015, we could see that the difference was that we could see that 89% were juice feeders or epistratic feeders were dominant group, whereas the rest of it were contributed by selective deposit feeders, which are particulate feeding, garbage collectors 1.13, and 0.87 predators were not noticeable. Epistratic feeders definitely dominated the outer fjord, whereas the predators were dominated in the inner fjord. Deposit feeders were distributed throughout the fjord. Of course, Dornopsis, Sabiteria were all epistratic feeders, were definitely dominant in the region. The deposit feeders, Tertilinga and Tertilinga, two communes and two species were dominant among the deposit feeders. Viscosia species is invariably a predator. Then the clavate and the most regarding the tail, if you look, tail shapes are the prevalent in the outer fjord, comparatively very low in the inner fjord, whereas the conical and filiform tails were common in both the fjords. Now, if I just look into the carbon potential of nematodes, you look, this was a study which has already been published. We look into the carbon strength. What is the carbon potential of the nematodes? How much the body of the organism is contributing to? We all know that around 4.4 into 10 raised to 20 nematodes are dominate the global. If you look into the global habitat, almost so much of nematodes occupy the surface of the soil. And the nematodes are very, very important in carbon structuring, in the recycling and bioturbation of particulate organic carbon, POC, and also particulate organic carbon. And the mineralization and remineralization of the carbon and the sequestration ability in distributing the minerals and of these meiofauna are notable, and we have already seen that it is very, very important. And POC, whatever is rarefied in the microbial food, that is also linked, and this, which goes to the residual heterotrophs, is also assimilated, and the important role done by nematodes is also very, very important. In this context, we calculated the carbon potential, how much it contributes to the Arctic ecosystem. We found that Anticoma herbertiva had contributed the highest carbon content of 14.06 microgram, followed by Meiolive complexa. Lowest value was recommended, was seen by Halalamus longicordatus. Total carbon contribution was very high, comparatively higher in the outer fjord. When the nematodes die and decay, the carbon definitely gets into the system and is sequestered into and we are also looking into the through our stable isotope and sequestration, we are looking into the potential, what is the source of this carbon and this potential in these Arctic fjords. This is another study which is parallelly going on. Now, if you look at 15, we will found that it's slightly different. What we see that Meiolive complexa, that's contributing around 12.94 of carbon and the highest carbon content, and the lowest was recorded by Tertilinga communis. Net carbon sequestered by the nematodes was the highest in the outer fjord and much lower in the. So the carbon sequestration studies indicated that the nematodes, as always among the meiofauna, are a key contributor to the carbon sequestration, which is a very, very important indication and potential and signature of the climate change scenario globally in any system, including the Arctic. Now, if I just want to take you to the foraminiferan studies of the 2011 period. If you look at the foraminiferan studies, foraminiferan distribution, 81% of the foraminifers are alive and remaining 19 were dead. Percent was dead, which we collected during the 2011 period. Abundance of foraminiferans was high in the outer fjord compared to the inner fjord. Overall, Nonulilla, Labradorica, and Cosidulina terris was most observed, which had a higher percentage was contributed by Nonulilla, and species of Nonulilla and another species of this Casidulina terris was high, and high organic matter were more prevalent in the outer fjord. And this is evident from these graphical representations which I have given, which shows the differences between the calcareous and agglutinated foraminiferan and in the two area. Suppose you look for 2015. If you look at 2015, 48% constituted by foraminiferal fauna, whereas 52% of the total population of foraminiferans were dead fauna. Outer fjord was always dominated by the calcareous foraminiferans. Middle fjord was dominated by the agglutinated forms. If you look at the abundance of foraminiferan was high in the outer fjord compared to the inner fjord. The faunal abundance of foraminiferans, overall the dominance of Nonulilla and also Cribioulphidium excavatum, followed by Cibicides species was also, and which contributed. Among them, the Nonulillas contributed the highest, that is 28.3, and we had the difference of the calcareous and the agglutinated forms. I have just given the in the middle fjord regions. Now, if you make a comparison, on the diversity during the Shannon diversity as well as the species richness and the evenness in the fjord for 2011, we could see the species richness and the Shannon index was low in the inner fjord for the Frams, as high, whereas it was higher for the outer fjord. The Piolo's evenness index and the species dominance did not show major variation between the two fjords. If you look at the multidimensional scaling ordination of the foraminiferal abundance, we could see that the cluster showed that the square transformed viridate, and the distribution of foraminiferans followed the two major clusters with overall similarity of just 30%. The NMD has also indicated the abundance of foraminiferans, that is 40% similarity in all outer fjord stations and 20% for the inner fjord, and with except for station 1. Yes, here you could see here. If you look for 2015, the difference was that higher abundance was observed in the outer fjord. The Shannon index, the species richness index was low in the inner fjord. Piolo's evenness index and the species dominance was also did not show major variation between the fjords. The NMD has indicated that the abundance of foraminiferans had 40% similarity between all the stations except station 1. The station 1, the low depth stations, the low 70, around 70, 75, showed 20% similarity with other stations. If you look into carbon potential, as I mentioned for nematodes, we could see that the cribrostomoids, Jeffersae, had the highest carbon potential, contributing to around 0.74 microgram, followed by Globulimina, auriculata, that contributed around 0.71 microgram. So the outer fjord had higher abundance and biomass of foraminiferans. If you look at the K plot or the K dominance plot or the curves, we had showed it was possibly high for all except for station 3, which is shown in light blue color here, where the S-shaped curve was evident, whereas the other did not provide the S-shape. This indicated that there was a stress condition, mostly because of the glacial inputs which is influencing the Arctic fjords. If you look at the 2015 carbon potential of these foraminiferans, we could see that Nonulilla labradorica contributed to the highest amount of biomass, that is 2.4826 microgram, as well as contributed a carbon of around 0.249 microgram carbon per meter, 10 centimeters square. So the K dominance plot here you could see on the right hand side showed the sloping curves were obtained for all the stations, which may indicate lower levels of stress compared to 2011 period. Now here also Nonulilla labradorica species was the dominant, abundant, possibly distributed around in all the stations. Its dominance was majorly explains the highly productive water zones associated with the warm inputs of incoming Atlantic water flow. as always, indicates the bloom diatoms as a food source was also being highlighted by many authors, Kurzum, Hall, Pollack, and so many more. Now, if you make a comparison, as I did for nematodes, we could see that 11 and 15, we made an overall comparison. The faunal abundance of Kong's Fjord was consisted of 56 species, whereas for 15, it was 43 species. The dominating calcareous species were Nonionella, then Casudilina, a couple of species. On the dominating species, we had calcareous species, Nonionella, then Cribroelphidium, then the Cibicidoides lobatius. Then we had other agglutinate species on the 2011. Other dominating species in the agglutinates were Rheophax to 2015. Then also Adarcotrima glomerchum, that is for 2015. Then less abundant, of course, was Dentalina and Ulina species, and also the less abundant here also it was Ulina and Spiroplectamina biformis. The abundance of agglutinated forms showed maximum density near the outer fjord around 2011, whereas the abundance of agglutinated forms showed the maximum density towards the inner fjords, where the depth was higher compared to the other stations. Monothalamus foraminiferans were rarely seen. It's notable, it's an important thing. It's notable during the 2011 studies, and monothalamus agglutinated foraminiferans were identified towards the inner fjord, especially Allogromia species. When you look at the physicochemical parameters, we could see that 2011 sediment samples, we correlated our data, the physicochemical, the principal component analysis was done. We could see that the inner fjord, and we made a comparison. Generally, the outer fjord and the inner fjord were compared. The pH variabilities was noticed in the pH, the granulometric analysis, sand, silt, and the clay fractions, the salinity, the total carbon, inorganic carbon, organic carbon in the inner and outer fjord. That is depicted here. I'll just come up and to the summing up. And because 2011 and this is 2015, we could see that from the 2011 and 2015, we did a principal component analysis. We found that the granulometric composition, moisture, pH, and the redox potential, oxidation reduction potential, and the total carbon, organic carbon, were the most important determinants driving the distribution of fauna, meiofauna in 2011. Whereas in 2015, the depth, salinity pattern, the moisture, total organic carbon, and this granulometric composition were the most important determinants determining the faunal distribution. This generally, we have identified more than 45 to 50 species of nematodes. I've just shown here the different species of forams. And now I'm just concluding the overall observations, what I made for 2011 and 2015. It's just summing up what the whole presentation, our whole study. Now, for 2011, the dominant taxon was Nematodes, contributing around 54. Summing up, for 2015 summer, the same period, forams and nematodes contributing 41% each. That's the difference. Species listed around 45 nematode species and 56 species of forams and other minor forms. Of course, we had 56 species of nematodes, 43 species of forams. Dominant species were Dorlonopsis, Tertilinca, Longicaudata Dorlonopsis contributing around 39.55. But here Dorlonopsis contributed around 63 during 2015, and Sabatiera about 15%. Dominant species of foraminiferans. Nonionella, you had forams 15%, Casudilina, then Casudilina, 2 species contributed to 2011. Here also, Nonionella contributed still higher, 28.30, and also Cibicides lobatus that contributed around 4.5%. Controlling factors regulating, the determining factors controlling the distribution and diversity of the meiofauna for 11 were mostly the granulometric composition, the moisture, pH, the redox potential, total organic carbon. Of course, the depth, salinity, moisture, total carbon, organic carbon, and of course, the sand, silt, and clay, the pattern in both the areas in the different locations based on the depth profile. Feeding guild analysis showed that majority of the species identified were deposit feeders and epistratic feeders for 2011, whereas epistratic feeders and deposit feeders were dominant for the 2015 period. Total biomass of meiofauna, we could see that the biomass of meiofauna was higher towards the outer fjord during the summer period of 2011, whereas for 15, the biomass of Meiofauna was higher towards the inner fjord region. Biomass in nematodes, whereas was higher towards the outer fjord, whereas that is for summer in 2011, whereas it was higher in towards the outer fjord for 2015. Biomass of foraminiferans, if you look, biomass was higher towards the outer, whereas that for forams also it was higher during 15. Abundance of Meiofauna, if you look for the general abundance of Meiofauna, it was around in the inner fjord was 57% for 11, whereas that for during the summer of 2015 it was 51% abundance. That is carbon potential, as I've already explained, it is Anticoma species, Meiolimnia species. These two are contributing the highest carbon potential, and from their individual bodies they contributed to the carbon in 2011. Meiolimnia alone contributed the highest, whereas along with other smaller species in 2015. Then of course the carbon potential of rams were of course contributed by cribrostomoids and also Globobilinimna and Nonionella for 2015, which I've already explained. But when you make a comparison on the time scale changes, what has already been reported from the previous studies, I'm just making the present study with the previous study, which has been already reported 2005, 2000, I think maximum we could get around 2005, 2004, then previous some studies has come from that. I'm just making a comparison on earlier study, we got reported a bottom temperature of around less or equal to 1 degree centigrade. Here we are getting around partially there's a rise in temperature in the water that is around greater than 3 degrees. We could see around 3 degrees. Salinity was around 34 to 34.7 to 35 ppd. Salinity here during the present study was around 33 to 34. Sediment texture, what was earlier reported by Hope et al. In 2002 was the sediments were dominated by muds. The content of sand was higher in the inner parts of the fjord. Sediment texture, what we observed during the present study, was dominated by the silt and clay in the fjord, suggesting a deposition of the water column is inputs from the freshwater runoff and from calving and ablation of the glaciers melting, because this is part of the global climate change phenomenon, and also the sea ice and the precipitation changes. Of course, the total organic carbon, the surface sediments of the fjord showed gradient with 0.04% to 1.7% towards the outer fjord. As far as total organic carbon by Shete et al., that's in 2011, he has reported. Total organic carbon, higher values of organic values were observed in the outer fjord, that's around 0.9 to 1.5% we reported. As far as Meiofauna was concerned, the number of the earlier studies have reported around 13 major meiofaunal groups, including Copipoda, Nopalea, Serpedia, Cypress, Parapludida larvae were recorded from the Kongsfjorden by West Lodarska and Kowalski et al. In 2016, whereas we reported the Kongsfjord glacial system, we recorded around 7 to 8 groups, including chinorhynchs, gastrotrix, copepods, harpicticoid copepods, polychaetes, bivalves, forams, nematodes, etc. Nematodes were dominated by Onchlemus and Thrist species. Of course, Onchlemus is present in low oxygen conditions. This was reported by Urban Malinga et al. In 2005, whereas also Dorolomopsis and Terslinga and Helilemus and Sagittaria was earlier reported by Sommerfeld in 2006. Of course, we reported Dorolomopsis dominant, Sagittaria and Terslinga almost going many species, but we have not reported Onchlemus this time from our studies. That's a very crucial, very indication. Similarly, for rams, dominant species were Elphidium excavatum, Casudilina, species of Nonionella labradorica, mainly dominated the outer fjord regions, less than 10%, that is reported by earlier in 2015, same time, earlier study or similar study, which was done by other authors. What we reported was definitely the dominance of Nonionella labradorica, then of course Cribro, Elphidium and excavatum. Then Nonionella labradorica dominated in all the fjords, and its abundance was about 25%. That is because in indication of the Atlantization of the Arctic waters. Now, just come to the sum up. I would like to acknowledge this particular work. This is part of a national work coordinated by the National Center for National Center for Polar and Oceanic Research, under the Ministry of Earth Sciences, Government of India, and they are coordinating this program. And I would like to thank for their coordination and the funding for conducting this work, which is still going on. We have been doing for the last 5 years. In the summer, we have been working in the Arctic Fox Fjord. Still very interesting results are emanating from our study. I would like to thank my researchers, Krishna Priya, Jima, Ashwati, Minu, Dr. Jayachandran, Akhilesh, all part of my research group who have been working in and out, and still my graduate students who are all part of this wonderful Arctic research program. Thank you all so much. Thank you. I've just skipped through some of our publications, which has already come through. It's for information, and of course, it can be shared, but still few are under review, and I hope we would get some more publications. Thank you all for a very patient. I hope I was audible and clear to all of you. Thank you, Jeroen. Thank you, Giovanni, all my colleagues on the Meolive program. Thank you all so much.